So, you've probably seen a lot of sensational headlines like this recently. Chinese lab crafts mutant COVID 19 strain with 100% kill rate in humanized mice! Oh no! But don't worry, calm down. No need to panic. This is actually something that is very common in research, and it's not unique to COVID. We do this with all sorts of different diseases like tuberculosis, the measles, HIV, hepatitis, etc. So first, let's dive into the article, and then we can talk about the journal paper. Throwing back in the memory banks, in a Wuhan-esque study, Chinese scientists are experimenting with a mutant COVID-19 strain that is 100% lethal to humanized mice. So when we looked at the paper later, the Wuhan-esque strain is they basically took the S strain variant, one of the original ones, and then they did a little bit of mutation to it to see what effects those changes would have on the disease and how it presents in the mice. Next, humanized mice. So we have something called HACE2 mice, which is a human analogous ACE2 receptor. We've done lots of different papers that use these types of mice. It's very common. They're not really humanized mice. They're just mice that have ACE2 receptors similar to us, so they respond similarly to diseases like COVID. And the name of this mutated COVID virus is GXP2V, which after 7 days had infected the lungs, bones, eyes, tracheas, and brains of the then dead mice, which caused severe enough disease to ultimately cause death in the animals due to the disease. And in the days before their deaths, the mice had quickly lost weight, exhibited a hunched posture, and moved extremely sluggishly. So why is that important? Well, that's very interesting because if we start to see these types of symptoms in people, that's like an oh no scenario. So if you have rapid weight loss, you start to develop like a hunched position, maybe because of pain, maybe because of something else, and you start moving very slowly, maybe like rapid onset ME/CFS could be something that would be like the diagnosis for that, then this might be an oh no scenario where you could have this fatal version of COVID. So let's go over the nonsense part of this article, where a supposed epidemiological expert at the University of London's Genetic Institute said this is terrible and scientifically totally pointless, and I can see nothing of vague interest that could be learned from force infecting a weird breed of humanized mice with a random virus. So this tells me that this person is either being 1, disingenuous, or 2, is really, really bad at their job. Because the reason why you would do this type of study and why it's very, very important is because if we start doing genetic sequencing of COVID and we start to see these types of mutations develop, we'll know, oh no, this is a very fatal strain of COVID, and that gives us a awareness, a heads up that if we see this specific genetic sequence or this type of genetic sequence, then we know that there is a very, very high death rate that will be associated with that version of COVID. Second, the behavioral findings in the study. If patients start to exhibit these type of symptoms, then we know, oh, this might be one of those really, really bad strains. So that gives us even more information. So 1, it's a surveillance heads up, like, hey, if you see this type of genetic sequence, this is an oh no scenario. If we start to see these types of symptoms prior to death, that's also a heads up for an oh no scenario. So this is very useful and valuable information. Next, the critique is from that same person. The preprint does not specify the biosafety level and biosafety precautions used for the research, and that this is madness that must be stopped before it's too late. Which again, this person is being incredibly disingenuous because COVID is classified as a biosafety level 3 disease, which means that you have to have these types of conditions to be able to run the study. Furthermore, when we look to this paper and we look at the affiliations of the authors, we see that there are hospitals, pharmacology labs, universities that are correlated with this study, and to be affiliated with a hospital or university, you have to have an ethics board review the work, and you have to have all these things, such as institutional review boards to monitor the animal research itself. There's absolutely no way that the biosafety level was not met for this to be done. And then also the fact that they're submitted it to a journal and it's a preprint, like literally, if they did not do that, they would be like attacked real hard, and this paper would not even be in preprint form. And so looking to the paper itself, we see that the lungs have very, very high expression and the brain has very, very high expression. So when we have this specific type of mutation, that GXP2V, it has a very, very high expression, and then you have the fatality at about 7 days. So what they did in this study is they did an injection into the trachea of the mice to inoculate them, and then they let it run its course, and by day 7, when the mice died and they did the autopsy, there was very, very high expression in the frontal lobes of these mice, as well as in the lungs. So these types of mutations of COVID, this specific genetic sequence, will result in rapid infiltration of the brain, which leads to these behavioral changes, deep lung infiltration, and then rapid death. So this is very, very important to know. And then to get an idea of what biosafety level 3 precautions are, I would recommend watching this video. It shows you how intense these types of precautions are and how many different areas you have to go through to be able to even get into the lab itself. There's lots and lots of phases. So any spillover risk would be, it's near impossible.